So indeed, today we're going to talk about how do we know if we've found aliens. And really the idea here is we're trying to look for aliens, which means beings that are alive on some extrasolar planet. So you're talking really, really far away. We can't send spacecraft, to go and take measurements. So the only thing we can do is look for the molecules in that atmosphere and then try and decipher whether ET or something is going on there that shouldn't be there. And so when we're looking for things, often we're looking for biosignatures. So a biosignature is a molecule that really shouldn't be present in an atmosphere at really high concentrations. So it's not going to be present in high concentrations because, one, it might not be formed by any natural processes. Two, it might react really quickly with the things that are going on. So for example, O2 in Earth's atmosphere, if there wasn't life continuously producing it, O2 would be destroyed by in reactions with other things in the atmosphere, which I'm sure Jenny could tell you a lot about. And so molecules that are rapidly destroyed in environments or aren't produced by natural sources very easily are good biosignatures. So O2, ozone are good biosignatures, and molecules that are just naturally occurring, like H2O, CO2, they're very bad biosignatures. So you'll hear a lot of papers in the news that are talking about, oh, we found water on some exoplanet. Water is a nice thing for life, but it definitely doesn't tell you that there's life there. And notice in all that definition, I didn't say anything about life producing it, because it turns out that on Earth, life produces pretty much any molecule you want. Geology, chemistry, photochemistry, natural processes without life are very limited in the number of chemicals that can be produced. So I was asked a few years ago what I thought the likelihood of us finding alien life in my lifetime, and this was my answer. Hopefully it'll come up. Probably 30 years we'll find something that looks like a biosignature, and then we'll spend at least the rest of my life arguing over whether or not it's a biosignature. And so what was really interesting in that clip in hindsight, oh, come on, next slide. You might have seen this molecule here in the corner. This is phosphine, which, 4 years after I took this video became obviously the big thing. So for those of you who aren't aware, in September 2020, an international team of astronomers announced that they'd found phosphine in the clouds of Venus. And the reason that was so important was because phosphine isn't produced in very high concentrations by geology. They actually have to try really hard to produce it naturally. And then the sulfuric acid in Venus should have just destroyed it really rapidly. So it's an oxidizing agent is what they call it. Oxidizing oxygen destroys it, sulfuric acid destroys it. It was actually a microwave line that was detected. So ALMA and these other telescopes detected the line. So of course that happened, and then there was lots of media articles. So first signs of alien life found on Venus. Life on Venus, stinky gas is alien made. So, these ones are going full hog, exactly what you want when you're being a very cautious scientist, being very considered, and someone tells you that you're claiming that you found aliens. This one's a much more sensible thing. Scientists find gas on Venus linked to life on Earth. That's exactly right. So Venus, they found phosphine. On Earth, it is definitely a biosignature. It's always associated with bacteria, or it's associated with human industrial processes. So they said for anomalous and unexplained chemistry on Venus. So this one's very sensible. I've actually written an article at about the same time, which I hope is sensible as well. So Anna's putting some things in the chat that you might want to look at and open later. And one of the things I remember saying when this discovery came about is everyone's like, oh, I don't think it's aliens, it's Venus, it's so hot. First of all, it's actually in the clouds, so it's actually a bit cooler, but there's so much sulfuric acid that, to be honest, it's probably not aliens. But you know what it is really good for? It's a really good test run for what happens when we detect something or think we've detected something that might be a biosignature. How do we go from there? What's the next step? How can we be sure what's going on? And so I want to take advantage of this format and go, how do we know it's phosphine? I'm just looking for a very simple answer here, a field starting with S that we all know and love. But I'd love everyone to find the chat function and just tell me, how do we know it's phosphine? Excellent. And you could have given them a little bit more time to figure it out. Yeah, more than one spectral line would be useful. You might think it's a pH vibration. It's actually one of the rotations that is going on. But look, the answer here is spectroscopy. And so what they did is, sorry, I didn't convert the units, but these are microwave spectroscopy units. They found a line. This, I think the black is their theory, the red is their model. So it's not the best signal in the world. You've got to use very, very high accuracy spectral data to be able to figure out what's going on. Then you want to look to see if there's a molecule that has the same spectral feature. So it turns out for this one, this is a pH 3 vibration, not vibration, rotation. But there's actually an SO2 rotation about, in these units, 1.4 away. So it's actually really quite close. So there's a line here and the astronomers are sitting there debating, is it phosphine? Is it just SO2 that hasn't been analysed properly? If you're wanting to know what's the latest, Anna's put it in the chat in terms of various people say it's probably not phosphine. The authors say, yes, we think it is. So I'll leave it to them to figure it out. The other thing that's come up is that the telescope processing might be incorrect. So it's quite challenging to go from, obviously, I think it's even more noisy than this, but very noisy data to your spectral data. That processing, they found some errors that were going on, but they've mostly corrected those and still think they've found a signal. Okay, so assuming for a moment, who knows whether it's phosphine, but assuming it is phosphine, how do we know it's life? And how would you figure out, as physical quantum chemists, spectroscopists, what do you think we need to do to know or its life or disprove? So I'll give you a moment and stare at everyone awkwardly so that they feel like they have to write something in chat. Send people, yeah, Terry's correct. The thing is that we can send people to Venus. Imagine if we were going to an exoplanet. We literally can't send people to an exoplanet. It is a bit sad. Yeah, so we've got model. We wouldn't know. It could just be a process that hasn't reached an equilibrium yet. So I think Matthew's sort of saying that that's a little bit what's happened on Mars, where they found methane and similar sort of discussions were going on and they didn't quite know what was happening. Proof by lack of imagination. Yeah, so we've actually got to do a full photochemical model and figure out what's going on. So I'll tell you what I thought when I heard this. I said that, well, okay, we've got phosphine. Let's look at whether there's other phosphorus-bearing molecules in the atmosphere. So I wanted to know what was producing the phosphorus and how was the phosphorus, what were the sinks, what were the sources and what were the sinks. So what else was going on in this atmosphere that we might be able to explore spectroscopically? Because remember, I'm interested in, I'd love to be able to send people to an exoplanet, but it's not going to happen. So how can we do it remotely? And on Venus, you're right, we can actually send a probe there, take some mass spec, figure out what's going on. Okay, so basically what I did when I had this question is I went and talked to everyone I knew, who, including many people in this audience, about what they knew about phosphorus molecules in the atmosphere. So I asked my PhD student Juan, who's down here. This is a very 2020 collaboration. some spectroscopic data people, microwave astronomers, experimental microwave spectroscopists, origin of life, atmospheric chemistry, kinetics, and enthusiastic undergrads, because we had some papers and some data to get through. So because this was obviously quite a high priority thing, we tried to do it as quick as we could. So the first approach I asked was, I said, okay, team, I want you to find every phosphorus containing molecule, which I called a P molecule, that might be of potential interest to Venus, that could tell us something about what's going on. And it turned out that was actually a much harder question, or more naive question than I anticipated. So the first question I asked is, which molecules are important in Earth's atmosphere? And Kieran and other people said, well, the master chemical mechanism and all these atmospheric models literally have no phosphorus in them. And I'm like, really? That's a bit strange. And it turned out if I'd done bio in my undergrad, I would have learnt about something called the phosphorus cycle, which basically tells you how phosphorus goes from, I'm not going to do a beautiful explanation of this, but it goes from, you know, the water, phosphates in the water, and then it goes into biology. So the dissolved phosphates go up to biology. Biology lives and then eventually dies. It goes back into the ground. It goes into biology, up, dies, round and round and round. But you'll notice in none of this is there any atmospheric involvement. So phosphorus-bearing molecules don't like to go in the atmosphere. So I said, well, okay, how are we going to figure out anything here? So luckily I had some origin of life and geology people, and I said, well, what do you guys know? There was some people talking about volcanoes, lightning. This original detection paper was telling us how they thought it might be formed. And it turns out that making phosphorus bioavailable as phosphates is crucial for life, but hard. So this is really to say phosphorus is essential to biology, and the biologists can tell you why, but it's really, really important for doing lots of things. But when you just have space and then you condense space into a rock to form a planet, then that phosphate is in, the phosphorus is in rocks. And if it's in rocks, it needs to get out of rocks into water so it can do something and get into eventually life and produce life. But getting the phosphorus from the rocks to the water is actually really, really hard. And you need things like lightning, volcanoes, extraterrestrial meteorites hitting something. These sort of processes have to happen. There's a few molecules that come out of this, but nothing very, there's not that many definitive molecules that come out of either of these processes. There's just like, oh, there's a handful of molecules that may be useful, but really we don't know exactly what's going on. So then I asked, well, okay, what high resolution data do we have for these molecules? So remember, if you've got no molecules, you literally can never detect, if you've got no data, the astronomers literally can never detect the molecule. So my next approach was, okay, stuff it. I don't care which one's important on Earth. I want any small organic molecule. And fortunately, someone had already done this sort of work before me. So there was a whole list of small molecules, and they basically said they're stable, did some things, and go, what do we think molecules might be present on an exoplanet that we might interpret as a biosignature? And they came up with 16,000 molecules, which is the topic of my one's PhD. And we basically said, got out the phosphorus ones. So we want to produce the spectra, predict the spectra. And previously you did it one molecule at a time. You said, I mean, many of you might have heard me talk about titanium oxide. Titanium oxide is really, really important. And I spent literally years collecting data for titanium oxide and giving the better data to the astronomers. But if we've got thousands of molecules, what do we do? So the first thing is we go, well, what would organic chemists say? So how would organic chemists predict the spectroscopy of small molecules? And I'll give you a moment to sort of think about this. And so organic chemists are really going to say, well, here's a molecule. What are the functional groups in that molecule? There's a CH stretch, there's a OH stretch, functional groups, bend, and then when we get to the whole molecule moving, we don't know what's going on. And that's basically what this researcher Clara Sousa Silva, who's actually, I mean, she's really involved in all this sort of stuff. She's called Dr. Phosphine because that was her PhD molecule. She figured out, she spent 3 years trying to find the spectra of phosphine really, really, really accurately, and then went to a postdoc and said, that's enough of one molecule. I want to do 16,000 molecules. Let's go. And she essentially made a computer program to do organic chemistry. So it divided the molecules into functional groups and constructed infrared spectra. So you can see on the left, around 3000, there's the CH peak. There's something that's gone a bit wrong with the OH peak, but you can see these different things that are going on. This is essentially a machine learning. Oh, actually, it's not really machine learning yet. We're hoping to get it to machine learning, but think organic chemistry done on a computer. Of course, I'm a quantum chemist, so I don't really know much organic chemistry. So I just use Schrodinger's equation, put Schrodinger's equation on big computer, solve the wave function, and just shut up and calculate. And basically, we use this phosphine detection as motivation to trial our approach, where we predicted the infrared and microwave spectra of phosphorus molecules as quickly as we could we were aiming to use fairly bulk standard techniques. So the sort of ones that you might teach, and you go, okay, can you use this for one of your molecules? And we're just going to do it for 1,000 molecules. So it is fast calculations. We're not going to try and dot all our I's and cross all our T's because we want to figure out what's important. It's no point going through the whole process, dotting it, like doing it absolutely perfectly and getting to the end and realizing a lot of those things you were spending a lot of time on were not important for the final answer. Yeah. Okay. So this is another picture of Juan. Juan really stepped up in this project. That's why I'm mentioning him so much. Anna is responsible for the chat, and she's done another great project, but that's not the one I'm speaking about today. But she's also responsible for a lot of the graphics, or at least teaching Juan how to do a lot of the graphics. So let's look at this plot. Amiga B97 XD Def2 SVPD, fairly bulk standard. We're doing harmonic frequencies. We do all our molecules. We put them in a computer, see what happens. Rascal, probably some important things is CompChem has more molecular specificity. So you can go, well, instead of just a CH group, we actually go, well, what's the environment? And we've got to spread out there. We do predict the fingerprint region. Of course, we've got to ask about the accuracy of that, but we're definitely producing data, which is good. This is a comparison between the NIST spectra at the top, which is basically the experimental spectra. And I've just noticed a question in the chat. The data on phosphorus molecules available experimentally is extremely, extremely sparse. So I don't know exactly, but like 5 maybe phosphorus molecules have spectra that you can easily access. And remember, we're talking with astronomers. If you can't easily access it, it's useless. So Rascal is this blue. So that's like the dirty organic chemistry approach. And quantum chemistry does better for this molecule, which is good for our last quantum chemists. We go, yes, we've got a job that is useful. These are a bit hard to do for a lot of molecules because there's not much experimental data. Oh, okay, well, my animations didn't work, but never mind. The questions we had naturally. So we've analyzed that, we've published that, I think the paper's in the chat. But really, I wanted to know, what is the best model chemistry? So we picked the one we did because it was reasonable, it had good dipole moments, and we'd benchmarked that, but we hadn't benchmarked the vibrational frequencies. And that's what we'd like to do nowish. And what errors can you expect? And it turns out, this is still very early stages, but the harmonic approximation itself, so when you use the harmonic approximation to predict fundamental frequencies and you scale them, so there's that scaling factor, and that'll get better. But that harmonic approximation, we think, has errors of about 25 wave numbers. So it's in one of those scaling frequency papers that come out, but it doesn't seem to be that widely appreciated. And I'd actually really like to talk to people afterwards, because I know they've been around this field a bit. Is 25 wave numbers sort of the best anyone can expect? Is that well known? Are we still figuring it out? There doesn't seem to be a lot of benchmarking of which model chemistry. So I'm looking for something like Lars's papers that go, this is the best DFT method to use, and it will give you errors of such and such for atomization energies. Those sort of things is what we want to get to, and I don't think we're there yet. 25 wave numbers, we're also not very happy with. So the obvious approach, if you're familiar with this, is to use perturbation theory. So generalized vibrational perturbation theory 2, so sort of like MP2 for vibrations. So this is the anharmonic correction. It should be better. Let me take you through this plot because it gets a bit complicated. The XMOL data that Clara spent years producing is down the bottom. That's the red data, and that's got all this rotational profile. It's beautiful. The scaled harmonics, so that's like just a really simple one we did before. Oh, whoops, go back. That's here in the black. So, you know, it's reasonable. And I think it's this one as well. Gray, rather. The black here, sorry, I don't know if you can see my mouse, but anyway, the black at 3,600. So it's taken a vibration that should be at 2,400 and put it all the way up to 3,600. So that's just what happens when you press the button on whatever program you're using. If you remove symmetry, it somehow reduces the error by 1,000 wave numbers because pH3, who knows what was going on. But it's still 500 wave numbers off. It's just completely unacceptable. Turns out if you do different levels of theory or Hartree-Fock even, you get much better results. And it turns out that this Omega B97X family has some problems with generalized GVPT2 that was recently found. So that's something we're going to have to explore a bit more. It's probably the higher order derivatives don't go very well with this family, but we obviously need to fix it. Oh, and this is just phosphine, but we actually found very similar results throughout the lot of it. So the weird thing about the anharmonic data was most of it was beautiful and you trusted it, and it was just a small correction to the harmonic and we're like, okay, pretty good. And then sometimes, mostly the pH stretches for some reason, were just off by 500 wave numbers randomly. So we'll have to figure out what's happening there. Of course, we're missing conformers here, we're missing isotopes, we're missing rotational broadening. They're all problems we're going to have a look at. There's definitely approaches that we can do. I will just say here, the resonances is part of that, but the generalized should actually deal with the resonances. So it's not just the symmetric one that has problems. It's just this is the one that was easiest to analyze. We're going to do some work on microwave spectral data. So this big data approach, very early stages, but basically we can figure out the dipole moments, which tells you the intensity, and that tells you the detectability of some molecules, which is kind of cool. And our data is useful because data is better than no data. And really it's about highlighting ambiguities for astronomers and preventing misassignments. So an astronomer, if you just give them data for water and they're looking in a certain region of the spectra, if it's actually ethanol or methanol, they'll go, oh no, it looks like water. It's got an OH stretch. Aren't we happy? And you're like, no, wrong. We need to give that data to astronomers because they don't know chemistry. The intensities of our data are reliable, so it can be used to identify strong absorbers that are likely to be observable. So we're trying to figure out which molecules are easiest to find. Machine learning is something another student of mine's working on. So whether we can do away with quantum chemistry, I don't think quite yet, but who knows? And really we're going to go from sort of a big data approach for lots of molecules and then get a higher accuracy approach, get into the lab, get into the astronomy observations, and sort of start this big data approach that I don't think has really been done much in vibrational spectroscopy. So I know I'm running out of time, just quickly. When you're trying to find aliens, spectroscopy is the only tool, assuming it's out of our solar system. And we need to connect all of these different fields and connect them to what you can observe. So astrobiology is a field that you may or may not have much time for, but sometimes what they fail to do is connect it to what can we observe? What is the effect of whatever process you're doing on the atmosphere? Because the atmosphere is the only thing we're realistically going to be able to look at. High throughput data production is really useful. but benchmarking the best approach will need some work. So that high throughput data production. And I'm just going to leave you with this slide and open up for questions. Sorry, Tyke, you've probably been screaming at me for ages.
Thank you very much, Laura. And
thank you, everyone.
So we have already got a few questions. Yeah, here we go, Matthew. Why don't you unmute yourself and go for it?
Okay, so I was just going to ask about the basis set you use for the phosphorus atom. Did you use the extra diffuse functions, or did you just use the whatever the base?
Yeah, we would have just used whatever the base is. It actually, it's a good point. I know we were chatting last week about it. We should think to improve that.
Yeah.
I think the benchmarking is what we're trying to do next, just knowing.
Yeah, I think the second row elements, as far as I'm aware, they need them, but I'm not an expert on this.
The other thing I think, though we haven't proved, the diffuse functions are less important for vibrational frequencies than for dipole moments. But yeah.
It could be also partly because I'm looking at complexes as opposed to just discrete molecules. Maybe that's why they're more important for me. I don't know.
That's definitely true, because the complexes will be more weakly bound, which means the diffuse functions are more important. But I mean, to be honest, we need to do proper benchmarking and figure it out.
I have a more general question actually for both of you speakers today, because we've heard about your work, and your work is an essential piece and a very big piece of picture here. So question number one is, for the students who are listening in and who will perhaps work in an area like yours. So how did you form your network to be this small piece of puzzle in this big piece of picture here?
Well, actually, I can give an answer to that one that might be useful. Is I was talking to Evan at this webinar series last year, and I'd sort of seen Evan, and I knew Chris Medcraft from the group he's at my university. Knew people from bits and pieces of conferences, and then decided last year to just go, okay, this really exciting things happened. What can we do? And I got meetings together and went, what do you think we can do, everyone? No, what really do you think we can do? And kind of pushed until everyone said something useful. And it was really useful and really exciting. And I learned a lot. Yeah. Take advantage of the people that you know a little bit and just try and see whether you can work with them. And if you can work with them, go for it.
All right.
Yeah.
And Jenny?
Yeah, I can add. I mean, definitely conferences, workshops, things. I was lucky enough to be in the model that I've used since my first year of my PhD has a workshop every year that's always prioritized students giving talks and student attendance, and that really helps to get to know the people in that community. But I think my field is also just fundamentally really open and collaborative, and I think it's because we can't really do that much with our own individual pieces. And so like the bulk of the co-authors on that project that I talked about were people I didn't know initially, but I went into the data archive and I said, who's measured these things before? Oh, this person. I'm going to send them an email and invite them to work with me. I'm using their data. And in my field, that's pretty normal. And we have massive co-author lists usually. So I think, I've had a lot of success from just kind of reaching out to people and saying, hey, I'd like to do this. I'd like to involve you. Here's what I would do. You'd be a co-author. What do you think? And people have usually jumped on board.
Yeah, that's excellent. I think, yes. It's good to hear that, for instance, in Laura's case, you actually have formed some network as a result of having the webinar series.
I thought it was pretty good advertising, but actually true.
Jenny, you have a question for Laura. Go for it.
Oh, yes, it's a very general question, but I want to repose the question that you started your talk with. So 4 or 5 years later, how likely do you think it is that you will find real clear signs of extraterrestrial life in your lifetime?
I think I've got the same, 30 years type thing, but what I didn't realize is how quickly they disprove it or argue about it Like it just happened so fast, all these people disputing it. I think it went from oh, this is amazing to I don't even know if it's happening in like 3 months. Yeah, so I don't know. I think everyone will just argue a lot more than I hoped. It's a bit sad, but that's okay.
It's a very, very significant problem and very big problem, and
Yeah.
undoubtedly.
Well, and I guess the other
strongly about it.
Well, and I think the other thing is that, look, you talk about aliens and you talk about extraterrestrial life because it's the one that you can tell your mum and dad and they understand. It's not like it's the only application of this sort of stuff. So, this sort of big data, once it's produced, if we can figure out how to use this big data to train a machine learning model to be able to take a molecule, predict vibrational spectra, then we can do that for all sorts of molecules, and they have all sorts of applications. And yeah, I mean, I guess a related question actually, Jenny, that I might be a good time to ask is spectroscopy, when you're talking exoplanets, so planets outside our solar system, is the only way you can get any information whatsoever. How important is it on Earth? Not really much at all, or in specialized applications?
There is quite a lot of spectroscopy behind the observations that my colleagues make. So we've got a brand new Fourier transform infrared spectrometer going on the roof of the New Molecular Horizons building here to complement the one that's been on the roof of the to replace the one that's been on the roof of chemistry since 1995. So many of our trace gas observations come from spectroscopy at some level. In terms of novel spectroscopy, like the kind of stuff that you're doing, I actually really don't know. I'm probably not the best person to answer that. I know people are making all sorts of cool new measurements of new species that we hadn't been able to detect in the atmosphere before. I actually don't know how most of those measurements are made.
And I guess we just keep on. The other thing I guess you have to do, Tack, with the interdisciplinary stuff is you just keep asking until you figure out where you fit.
That's right. You get really comfortable sounding really stupid and ask questions to people who, and it takes a while to learn to understand each other.
When they told me that there was no phosphorus, they came back and were telling me how phosphorus came to Earth, and I'm like, but it was just there. Like, you build your planet, it's there, and they're like, oh no, it's not because it's locked in the rocks. And that took me a while, and then I'm like, oh, well, that's kind of fun.